Pain seminars with Module 7 Peripheral and Central Sensitization. In previous modules, we have discussed the differences between acute and chronic pain on different occasions. Acute pain, as in this picture, when you attempt to cut onions but actually slice part of your finger. Acute nociceptive pain is a physiological sensation of hurt that results from the activation of nociceptive pathways by peripheral stimuli of sufficient intensity to lead to or to threatened tissue damage. That's known as nociceptive stimuli or noxious stimuli. In this module, we will look at sensitization from a slightly different perspective and go a little bit more in depth. Sensitization can be seen as a generator of pain hypersensitivity by central neuroplasticity. As we have seen in previous modules already, that goes together with increased membrane excitability, increased synaptic efficacy, and a reduced inhibition pattern. We've looked at that in one of the previous modules. So sensitization can be seen as an enhancement in the functions of neurons and circuits in nociceptive pathways caused by 1. An increase in membrane excitability, 2. An increase in synaptic efficacy, and 3. As a result of reduced inhibition. When you first start studying the differences between central and peripheral sensitization, they may appear quite similar. However, on closer look you will see that they differ substantially, both in terms of the molecular mechanisms responsible and their manifestations. So central sensitization is not the same as peripheral sensitization. In this animation you see how nociceptive input coming from the periphery, on the right side of your screen, reaches the dorsal root ganglion where the C fibers release chemicals in response to the nociceptive input. These chemicals go prodromically direction dorsal horn where they activate certain neurons. If the stimulus is strong enough or the frequency is intense enough, it will actually trigger a signal to the contralateral spinothalamic tract amongst others. The chemicals also are released antidromically or against the flow back towards the periphery. And the release of those chemicals back into the periphery is shown in this animation where indeed multiple chemicals are released in not just the skin but also potentially in muscles. Many of these chemicals were later identified to be playing a role in myofascial pain. So if we look at what happens in the periphery first, it's fair to say that peripheral sensitization represents 1. A reduction in the threshold, and 2. An amplification in the responsiveness of nociceptors. So with peripheral sensitization, it is thought that the peripheral terminals of normally high threshold primary sensory neurons are exposed to these inflammatory mediators we just saw and to damaged tissue. In other words, peripheral sensitization contributes to the sensitization of the nociceptive system and to inflammatory pain hypersensitivity at these inflamed sites. And as such, it represents a form of pain elicited by activation of nociceptors in the periphery. Let's take a look at a brief video about the phases of nociceptive pain.
Nociceptive pain. Nociceptive pain occurs as a result of the activation of the nociceptive system by noxious stimuli, inflammation, or disease. Nociceptive pain proceeds through 5 phases: transduction, conduction, transmission, perception, and modulation. Nociceptive pain originates in the nociceptors, which are highly specialized primary sensory neurons found primarily in the skin, or joints, or the walls of organs. These receptors convert noxious stimuli into electrical current through a process called transduction. The current is then conducted along the nociceptor axons to their cell bodies, located in the dorsal root ganglion in the spinal cord, and then to their central terminals located in the dorsal horn. Here, the electrical impulses initiate neurotransmitter release from nociceptor central terminals, which relay the signal across synapses to the dorsal horn neurons. Greater stimulus intensities are associated with the release of neuropeptides, including substance P, from central terminals of C fibers. Substance P, acting via neurokinin receptors located on dorsal horn neurons, generates a greater postsynaptic response and enhances the activity of NMDA receptors. The signal is then relayed via ascending nociceptive pathways to higher centers in the brain, where it is perceived as pain. Ascending nociceptive transmissions are modulated by inhibitory or facilitatory control systems in the dorsal horn of the spinal cord. The process of inhibition and facilitation is regulated by neurons projecting from the nucleus raphe magnus in the lower brainstem. Nociceptive signals are transmitted through descending projections in the dorsal horn of the spinal cord. Facilitation serves to provide warning of tissue injury and encourages protective behaviors such as fight or flight responses, while inhibition may serve to provide analgesia at times of danger so that pain does not compromise function.
While this video is quite accurate, it should be noted that when the voiceover said that nociceptors come mostly from skin, joint and the walls of organs, of course he did not mention that muscles, and for that matter nerves, also have nociceptors, which is much more relevant for the myofascial pain story. As Dr. Kathleen Sluka has pointed out on her private website, there really are two schools of thought regarding peripheral and central sensitization. The more traditional school of thought believes that once pain becomes chronic, the peripheral injury is healed and nociceptors therefore contribute little or not at all to the ongoing pain experience. A new modern school believes that all pain has a nociceptor that is activated and involved in maintaining the pain. Dr. Hong-Yu Ga feels that central sensitization is not always presented properly to clinicians and may at times be overstated and overestimated. He does believe that it is important, as is the realization that it is maintained by peripheral nociceptive input. In the previous slide, we saw that peripheral terminals of high threshold primary sensory neurons can be exposed to either inflammatory mediators or damaged tissue, which is one model that is more consistent with one of the schools of thought. There is, however, a third option based on more current literature that you can have peripheral sensitization without these inflammatory mediators and without tissue damage. For some of the opponents of the myofascial pain construct, this is a concept that is not acceptable. And people believe that you cannot look at muscle that is not damaged and still talk about trigger points being sources of nociceptive peripheral input. Normally, pain that is produced by muscle activation or exercise produces mild, short-lived achiness or muscle soreness. We are familiar with that. This is present with lengthening contractions of muscle or eccentric contractions, supramaximal contractions, and also with repetitive low-grade contractions seen in the Cinderella hypothesis. The soreness may be greater a day or two after exercise, a phenomenon known as delayed onset of muscle soreness, or DOMS. This model of DOMS has been taken sometimes as a model for hyperalgesic trigger points or myofascial pain in general. And there are actually several recent studies that show that even in a condition of central sensitization known as fibromyalgia, the pain can still be dependent on peripheral nociceptive input. And as a result, allodynia and hyperalgesia associated with fibromyalgia can be improved upon or maybe even abolished by removing the peripheral impulse input. Roland Stott, a professor in Florida, has published several papers on this subject. And the studies by Rubin from Australia also showed that the maintenance of referred muscle pain, which as you know is a feature of central sensitization, depends on ongoing noxious inputs from the site of primary muscle pain. There is also evidence from non-muscular sources of peripheral nociceptive input in a symptom like fibromyalgia. In this paper, they found small fiber pathology in patients with fibromyalgia syndrome, and what they really found was a reduction in the dermal unmyelated nerve fiber bundles in skin samples of patients with fibromyalgia compared with patients with depression and with healthy control subjects, whereas myelated nerve fibers were spared. And every method they used showed that in patients with fibromyalgia, there may be a neuropathic peripheral nature of pain. So even in these fibromyalgia patients with central sensitization, the pain may be caused or maintained by peripheral nociceptive input, in this case from dermal unmyelated nerve fiber bundles. Here is another paper that shows that peripheral nociceptive input can indeed contribute or maybe underlies some of the mechanisms of central sensitization of fibromyalgia. In this case, 41% of subjects, compared to 3% of control subjects without fibromyalgia, had small fiber peripheral neuropathy with a lower epidermal nerve fiber density. And as the title suggests, they felt that this small fiber polyneuropathy may underlie some of the illnesses currently labeled as fibromyalgia. Here's another study from the research lab from Roland Stott from Florida that shows that lidocaine injections increase local pain thresholds and decrease remote secondary heat hyperalgesia in fibromyalgia patients. In other words, peripheral impulse input played an important role in maintaining central sensitization. If we now focus our attention to myofascial trigger points, according to Hangyu Gar and colleagues in this article, indeed, active myofascial trigger points can be thought of as one of the major peripheral pain generators for regional and generalized musculoskeletal pain conditions. Recent evidence in the understanding of the pathophysiology of myofascial trigger points supports the integrated hypothesis for trigger point formation. Clearly, not everyone is in agreement with this, and there are quite a few people who say that trigger points are not peripheral pain generators, but they also then do not provide an explanation to the phenomenon that we have come to know as myofascial pain. So the local pain and tenderness at myofascial trigger points are largely due to nociceptor sensitization. That would be the case of active trigger points, with a lesser contribution from non-nociceptor sensitization, and that would be from your latent trigger points. This may in part be due to the process of muscle ischemia associated with sustained focal muscle contractions and or muscle cramps. Acute pain occurs with a temporal relationship to an injury or other activation stimulus like a distension of the bowel or bladder. It usually produces an expected pain and goes away when the injury heals or the stimulus is removed. Acute pain serves a protective function. It makes us remove our hand from the hot stove. Chronic pain or persistent pain, on the other hand, persists long after the injury has healed, after the stimulus has been removed. It involves more than the part originally injured. The pain is out of proportion to any identifiable stimulus. Chronic pain serves no protective function, and in fact it may become a disease in its own right, where the constellation of symptoms like fear of movement, withdrawal from family and friends, depression, and insomnia. The story of how acute pain becomes chronic pain is the story of sensitization of the peripheral and central nervous systems. Sensitization of the nervous system occurs rather quickly, often within days, of repeated or intense painful stimulation. We will describe how this comes about in the course of this lecture. Sensitization has three major consequences. It lowers the threshold required to activate nociception. That is, less severe or more minor stimulation will result in pain. It also amplifies the pain response so that painful stimulation becomes more painful than expected. You may compare that to a pat on the back of a sunburned person. And non-painful stimulation, like light touch, becomes painful. So this slide summarizes the mechanisms, the consequences. So it occurs after repeated or intense noxious stimuli. It lowers the thresholds for activation. It amplifies subsequent inputs, and it is adaptive. In this animation, you see how nociceptive input from the periphery enters the dorsal root ganglion, then comes into the dorsal horn with sufficient frequency and intensity. The signal will cross over in the contralateral spinothalamic tract most commonly, goes up into the brain, and there is an awareness of pain. In our course program, we talk primarily about peripheral nociceptive input from myofascial structures, but it is important to realize that very similar patterns occur with osteoarthritis and other pain sources. Here you see a similar diagram where pain from an osteoarthritic joint enters the dorsal root ganglion, the abbreviator is DRG, goes into the dorsal horn, crosses over in the spinothalamic tract and reaches the brain. This is a fairly recent review of a mechanism-based approach to pain management in osteoarthritis with many similarities to myofascial pain. As we already mentioned, the most common pathway is indeed the spinothalamic tract, but the body has a lot of duplication. There's also a spinal reticular tract and a spinal mesencephalic tract. These are different targets, different mechanisms, and we don't know much about it in the context of myofascial pain. So for our purposes, the most important one to remember is the spinothalamic tract. Studies by Nidham, who provoked myofascial trigger points in the upper trapezius muscle, showed that this kind of noxious input from the muscle projects to the rostral, ventromedial medulla, and the periaqueductal gray. The input in the dorsal horn is predominantly through the superficial lamina 1 and 2 and the deep lamina 4 and 5 in the spinal dorsal horn. You may recall that these lamina from Rexed, R-E-X-E-D, determine to a great extent what happens with the processing of input. When trigger points were stimulated, patients experienced significantly higher levels of pain, also known as hyperalgesia. The corresponding brain responses involved the enhanced somatosensory and limbic activity, and it suppressed the right dorsal hippocampal activity. So there are specific parts of the brain that get involved with peripheral nociceptive input from muscles. There is no question that repeated nociceptive input from muscle causes central changes in how these nociceptive inputs are being processed. In this study by Rubin from Australia, the subjects received a weekly injection of a noxious stimulus in the proximal anterior tibialis muscle. In the beginning, there was a local pain with some referred pain, but after 4 weeks, the local pain was pretty much diminished and the subjects only experienced referred pain farther down their leg. This suggests central changes in how noxious input from muscles is being processed. A few slides ago we mentioned already that musculoskeletal nociceptive input is processed through lamina 1, 2, and 4 and 5 of the spinal cord. In this simplified block diagram, on the left you see the peripheral input and going into the spinal cord, then the brainstem, the diencephalon, and then eventually the cortex, as indicated on top of the slide. Input from the musculoskeletal system is transferred through A delta and C nociceptive fibers and low threshold A beta fibers. You see here that the dashed lines represent inhibiting and therefore mostly descending pathways, the ultimate effect of which is an inhibition in lamina 2 of the spinal cord. The numbers 1 to 9 represent the corresponding laminae of rexed, R-E-X-E-D, of rexed of the spinal cord. We see that the information gets processed to the reticular formation, abbreviated as RF, the periaqueductal gray, abbreviated as PAG, the ventral basal nucleocomplex of the thalamus, abbreviated as VB, and the primary somatosensory cortex and secondary somatosensory cortex, abbreviated as S1 and S2. Here you see the lamina in a slightly different representation, so the dorsal horn neurons that receive nociceptive input from skeletal muscle are located in laminas 1, 2, 4, and 5 of the dorsal horn. Central sensitization occurs through the enhancement of normal processes of cellular activation. The dorsal horn neuron membrane becomes more excitable through changes that we'll discuss shortly. As a result, the synaptic connections between axonal nerve endings and the dorsal horn neuron become more efficient. So previous subthreshold synapses reach threshold. The result is the conversion of previously inactive synaptic connections become active. Thus the dorsal horn becomes responsive to areas of the body to which it had previously not responded. In addition, the descending inhibitory system becomes less effective, reducing the normal supraspinal descending nociceptive inhibitory control that modulates ascending nociceptive transmission. As we have stated already a few times, the descending inhibitory system becomes less effective, reducing the normal supraspinal descending nociceptive inhibitory control that modulates the ascending nociceptive transmission. So descending supraspinal control of the spinal nociception originates from several brain regions, and this control can be altered due to behavioral, emotional, and pathological states. A facilitating shift of the descending system has been reported for patients with persistent pain. So the disinhibition is a major mechanism for triggering and maintaining central sensitization. Sensitization causes local changes in the perception of stimuli and also more widespread response to a local stimulus. We recognize allodynia, hyperalgesia, and secondary hyperalgesia. A painful stimulus that is more painful than expected is called hyperalgesic, and it represents hypersensitization. Allodynia is when a normally painless stimulus is felt to be painful. Both hyperalgesia and allodynia can develop outside of the area of injury, which is a manifestation of central sensitization. Secondary hyperalgesia is a spread of pain beyond the site of stimulation of injury and this is clinically referred to as referred pain. To illustrate the concept of allodynia, we're turning to the graphic novels of Asterix and Obelix. See what happens as this leaf works its way down towards a sleeping individual. As you can see, there is a painful response to a non-painful stimulus. This would be considered allodynia. To display this in a graphic format, let's take a look at this graph of pain intensity versus stimulus intensity. Under normal circumstances, you need a certain degree of stimuli to have a pain response. However, in sensitization, this entire graph shifts to the left, meaning that you need less stimulus to get a pain response. And that applies both to allodynia and hyperalgesia. So central sensitization is the phenomenon that underlies many of the problems that we see frequently, such as neuropathic pain, inflammatory pain, migraine, IBS, fibromyalgia, and indeed myofascial pain syndrome. We realize that this information can be quite challenging for some of our students. This is typically not taught in physical therapy school or other schools of healthcare providers, and therefore we have built in a little bit of repetitiveness, just to make sure that you understand what the current thinking is about sensitization. Sensitization changes the function of sensory neurons in the dorsal root ganglion and in the dorsal horn of the spinal cord, as we have already seen. They can change high threshold A-beta mechanosensitive neurons to low threshold mechanosensitive neurons, for example. The pain pathways can be activated by wide-ranging responsive neurons that now respond to a greater array of stimuli than normal. The result is activation of nociceptive pathways by minor or usually innocuous stimulation. Nociceptive pathways can be activated in the absence of any peripheral stimulation once sensitization has occurred. Perhaps the following cartoon will explain the process of central sensitization even better.
Under normal physiologic conditions, light touch is transmitted from low threshold receptors in the skin through large A-beta fibers to a network of neurons in the spinal cord. The signals are transmitted along this low threshold pathway to sensory centers in the brain. Nociceptive pathways conveying signals from high threshold mechanoreceptors in the skin include a distinct pathway that passes through neurons in lamina 1 of the spinal cord. However, nociceptive and light touch pathways are interconnected. They communicate through a complex network of synaptic connections that include inhibitory interneurons in the dorsal horn. These inhibitory interneurons normally act to repress the flow of nociceptive-related information, and in addition, mask the interconnected nature of low-threshold and nociceptive pathways. Thus, light touch produces no excitation of nociceptive pathways, and in fact, can even serve to repress them. However, when the control signal from these inhibitory interneurons becomes less efficient or is inverted, the excitatory transmission between low-threshold and nociceptive pathways becomes possible, and this crosstalk presents clinically as tactile allodynia.
The information from the previous video clip is summarized in this picture of normal sensitization. At the bottom part of the slide you see tactile information transmitted by low threshold mechanoreceptors experienced as touch. On the top of the screen you see nociceptive input processed by nociceptive fibers and experienced as pain. As you saw in the video, there are connections between the two systems. So in the case of central sensitization, the low threshold mechanical input at the bottom of the slide can actually cross over to the nociceptive part of the system and this experiences pain, which we then call allodynia. The touch sensation is diminished dramatically as pain has taken over. At the same time, nociceptive input in the top part of the slide is experienced as a much stronger signal, that which is referred to as hyperalgesia. In the normal situation, a single dorsal horn neuron gets information from, or perhaps you want to say, sees a particular region of the body that is called its receptive field. Activation of the cell is normally produced by stimulation from nerve axons from this receptive field that belongs to that neuron. Each neuron has many synaptic connections, numbering into the hundreds of thousands, and coming from other regions that are not the primary receptive field of the neuron, but that normally do not generate sufficient activity to reach thresholds sufficient to depolarize the cell membrane. So the information that may come from outside its own receptive field is partially the result of a degree of overlap of receptive fields. This slide illustrates that receptive fields indeed overlap with other receptive fields, meaning that neurons can receive information from other regions. Under normal circumstances, the threshold is not reached to activate neurons when information from other regions enters the cell. Here you see another representation how one neuron can indeed receive information from other receptive fields other than its own primary receptive field. By now, this slide should look familiar. We've reviewed it before in less detail. Here you see the classic experiment by Siegfried Menzi from Germany, in which they placed recording electrodes at neurons in the dorsal horn of this rat. Before they did the experiment, they established the receptive fields of various neurons. So in this illustration, here is the receptive field of the neuron at L3. But they also established receptive fields at L4 and L5, etc. Then they created an inflammatory condition by injecting, for example, bradykinin in one or two of the muscles of the limb. They identified the receptive field of a dorsal horn neuron by stimulating the hind leg muscle of the rat with a strong pinch, until the region that had significant electrical discharge from that recorded cell was established. What you see here, that before the injection of bradykinin, they did indeed need a deep noxious stimulation to activate the neuron in the dorsal horn. But just after 5 minutes, they noted that the neuron at L3 now also received information from the receptive fields that before belonged primarily to L4 and L5 neurons. And a few more minutes after that, not only was there an expansion of the receptive field, but there was also a level of sensitization. It did no longer require a deep noxious stimulation to activate the neuron at L3. In other words, the threshold for stimulation of L3 was reduced as a moderate technique of pinching would activate the neuron. The equivalent of an expansion of the receptive field of the neuron is what we call tenderness, which really is nothing more than a reduction in the intensity of the stimulus required to elicit a response from the neuron. So in summary, each neuron has a primary receptive field from which it receives a stimulus from a single peripheral nerve cell, whose cell body is in the dorsal root ganglion. The receptor of that peripheral nerve is in the muscle, in this case in the hind leg muscle of a rat. The shorter axon of the peripheral nerve extends from the dorsal root ganglion nerve cell to the dorsal horn of the spinal cord, where it forms a synaptic connection with the dorsal horn neuron. That pathway is illustrated on the left side of the cartoon. However, a single dorsal horn neuron does not receive input from only one incoming nerve axon. In the right side of the cartoon, the two additional receptor fields each have a primary dorsal root ganglion nerve cell that has a primary input to another dorsal horn nerve cell. But each of these dorsal root ganglion nerve cells also sends an axon to the first dorsal root ganglion. That's the one whose activation we are recording, let's say the L3. In the illustration, this happens by a branching of the central, incoming, or afferent axon. In reality, each dorsal nerve cell is estimated to have about 2 million synaptic connections. In other words, there is extensive branching of the incoming afferent axonal tree, which branches extending far up and down the spinal cord. The dotted lines of the branching axons indicate that these connections are usually ineffective. That means that the intensity of the stimulus they present to the nerve cell is weak, and too weak that the stimulus cannot activate the nerve cell. It cannot depolarize the membrane and initiate an action potential. We call such a stimulus a subthreshold stimulus. When these ineffective synaptic connections become effective, then the receiving dorsal horn neuron sees the area from the nerve axons arises, from the peripheral area in muscles that they innervate. The mechanisms that underlies this change is an increase in the synaptic efficiency of the axonal connection to the dorsal horn neuron and the change in the nerve cell that allows this to happen, which is called neuroplasticity. To experience muscle pain, there are two activating chemical substances of particular importance: adenosine triphosphate, or ATP, and protons, which are linked to a drop in tissue pH. These chemical substances activate nerve endings by binding to receptor molecules located in the membrane of the nerve ending. ATP activates mostly muscle nociceptors by binding to the P2X3 or other variations of this receptor molecule. Protons bind to the receptor molecules of the transient receptor potential vanilloid 1 and acid sensing ion channels. The receptor molecules are channel proteins that span the membrane of the nerve ending and mainly permit sodium ions to enter the neuron, which induce neural excitation. The exact mechanisms why latent trigger points do not elicit spontaneous pain but can evoke referred pain are really not known. In the article How Do Muscle Lesions Such as Latent and Active Trigger Points Influence Central Nociceptive Neurons, Dr. Siegfried Menzi speculates that pain referral from a latent trigger point is probably due to the fact that latent trigger points only have ineffective connections with the central nervous system, and that these synapses are located on neurons that supply regions remote from the trigger point. As you see in this cartoon from this article, there must be many different chemicals involved in the dorsal horn itself to facilitate these pathways. You see, for example, substance P, calcitonin gene-related peptide, and brain-derived neurotrophic factor, or BDNF. BDNF is a very interesting chemical because it facilitates better synaptic connections. It is, for example, activated after aerobic activity. And there's some research that suggests that if you have a difficult exam, let's say for example the Pain Seminars exam, it would make sense to go running or do high aerobic activity about an hour before the exam, as it will release significant amounts of BDNF, making you actually more efficient in your thought processes and therefore probably leading to better results on your theoretical exam. Just a thought. I can imagine that by now your head is spinning a little bit from all this information. So let's take another look at the mechanisms of referred pain. The increase in cell membrane excitability allows the normally subthreshold impulse to the cell, the so-called sleeping connections, to reach threshold and activate the cell. So you need a recruitment of subthreshold inputs. You get an activation of the nociceptive neuron. And some things we haven't talked about is the role of NMDA receptors, or the N-methyl D-aspartate receptor, which mediates also hypersensitivity. We are going to talk about the NMDA receptor in more detail in the next module. In addition, there is a loss of tonic inhibitory control known as disinhibition. And sometimes we haven't talked about yet are glial neuronal interactions. You have learned by now that the changes in the receptor field properties have a threshold component, a spatial component, and a temporal component. The sensitized neuron, which may have had a high threshold to mechanical stimulation, now responds to low-intensity mechanical stimulation as well. Neurons that once only responded to noxious stimuli respond now to innoxious stimuli, and by doing so they become so-called wide dynamic range neurons. There's some confusion about the phenomenon of wind-up. Wind-up produces an increasing response to continuous stimulation, but it ends when the stimulation ends, and therefore it's really not a manifestation of neuronal plasticity. It is now known that many structures of the central nervous system can exhibit central sensitization, as represented in the schematic presentation here. The first evidence for central sensitization was generated in 1983 by revealing injury-induced changes in the cutaneous receptor field properties of flexor motor neurons as an integrated measure of the functional plasticity of the spinal cord. Conditioning noxious stimulation resulted in long-lasting reductions in the threshold and an expansion of the receptor field of the motor neurons that was shown to be centrally mediated and generated. Similar changes were then described in lamina 1 and lamina 5 neurons in the dorsal horn of the spinal cord, but also in spinal nucleus pars caudalis and other brain structures such as the thalamus, the amygdala, and the anterior cingulate cortex. There are many imaging studies now that have revealed several brain structures in human subjects that exhibit changes compatible with central sensitization, in this diagram represented by the blue dots. So many of the pain-generating structures in the body, whether it's from muscle, joints, or others, present similarly at the central nervous system level. Again, the changes are not confined to the dorsal horn of the spinal cord, and they occur in the brain as well. The nucleus caudalis, or caudal-most nucleus of the trigeminal nerve, is really a continuation of the dorsal horn in the brainstem. The thalamus, the amygdala, and the cingulate gyrus are projection areas that receive input from dorsal horn nociceptive neurons. As you already expected, the story is in fact much more complicated than we make you believe. In patients with chronic pain conditions, for example, there are alterations in the connectivity, the morphological and biochemical changes in the pain matrix. It's been shown for many chronic pain conditions, including fibromyalgia, where there's a decreased intrinsic brain connectivity associated with reduced clinical pain. There is some confusion why dry needling or manual trigger point techniques would not cause central sensitization. Well, this is mostly due because to get central sensitization, the noxious stimulus must be intense, repeated, and sustained. And the input for many fibers is required over tenths of seconds. So a single stimulus, such as a pinch or a needle stick, is insufficient to induce central sensitization. It's also important that tissue injury is not necessary for central sensitization. Especially in the manual therapy world, you hear frequently that in the presence of central sensitization, physical therapy or other therapies must be pain-free to avoid creating more central sensitization. But again, it seems that the short duration of our nociceptive input from dry needling or manual trigger point techniques is too short to induce central sensitization. Along those lines, there is emerging research that shows that if we educate a patient that the noxious stimulation, for example, from dry needling, which in fact is a conditioning stimulation, can actually lead to a reduction in pain, it will actually lead to a larger inhibitory conditioned pain modulation. And this procedure can activate an endogenous pain inhibitory mechanism that inhibits early nociceptive processing. So the inhibition of one source of pain by a second noxious stimulus, be it manual technique or a dry needling technique, is termed a conditioning stimulus. And the pain intensity can be dissociated from the magnitude of responses in the pain matrix. The responses in the pain matrix are strongly influenced by the context within which the nociceptive stimuli appear. So if it's clear in your therapeutic encounter that you are working hard to reduce the patient's pain, this painful stimulus of a pinch or a manual technique or a dry needling stimulation, which in other circumstances would have been experienced as painful, does not trigger central sensitization. It is quite interesting that although needles are often thought to be a threatening tool and a threatening nociceptive input, as expressed here, for example, on the somasimple.com website, very few people actually suffer from trypanophobia or an excessive fear of needles. Patients who would suffer from trypanophobia would probably not come to clinics that advertise that they use dry needling techniques or injection techniques. So I think it's fair to assume that the patients who do come to clinicians who use painful manual techniques or invasive dry needling techniques or injections do not suffer from trypanophobia. And that fear of needles that many people have to some degree really has not been found to be a reason not to be effective in your therapies, whether it's manual therapy or instrument-assisted manual therapy, known as dry needling. So a fairly recent study showed that fear of needles does not influence the pain tolerance and sympathetic responses among patients during a therapeutic needling session. And I think this has to do with the conditioned pain modulation response, as we discussed earlier. So, although people often believe that painful therapies should be avoided in chronic pain patients, the research actually shows quite the contrary. Painful manual techniques or painful dry needling techniques have very little influence on the pain tolerance and actually in most cases immediately reduce the pain by starting this endogenous pain inhibitory system. This concludes the first lecture on peripheral and central sensitization. I can imagine that some of the information was quite challenging, so therefore we recommend that you review this module several times and that you read the articles that accompany the text and to get to a better understanding. It is a critical part of the equation to explain, to understand why patients develop sensitization, what the mechanisms are, and eventually what you can do about it. And that brings